I started the recording early again, so if you're watching the video, the first word for attendance is Friday, and you can fast forward. Let me see if I can fix this camera situation. Bear with me. It's always something, right? Well, yep, cannot zoom in or out with the camera. So, yeah, I guess once you see me share the PowerPoint, then you should stop fast forwarding. Morning. So we got a handful of people here early. The first word for attendance will be, well, obviously for you guys it's extra credit, but for those of you online, it'll also be extra credit. And let's see, the first word is eraser. And as always, is a good reminder. If you send the words in and I don't give you the extra credit, remind me that that word was extra credit and not just one of the regular words. All right. This is also another reminder, since spring break just ended, getting back into the swing of things. So remember, if you're online, you still have to send the words in, right? So just because you've logged on doesn't mean you're here. You get the points. Because as I'm logging you down, for those of you who are online, I'm seeing some names who have not sent words. Like you've been here online, but you haven't sent words. So make sure you email me the words after we're done, sometime before 9 a.m., right? The sooner the better, but sometime before 9 a.m. would be good. Again, just because you are logged on does not mean you're getting the points. Please make sure you send the words. Morning. 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 Let's see here. Morning. So Hunter, right? No, not Hunter. Yes, Hunter. Yeah. Hunter's here. Caleb's here. Let's see. Is it Canterbury? No. Jones? Crowder. Yeah. It's been a week. That's where I'm making the mistake. Jones? Tate is here. All right, it's midterms. So do some reminders since we are halfway through the semester. And I already did one of the reminders for those of you online. I'll remind you again. Just because you've logged on online doesn't mean you have the attendance points. You have to send me the words before 9 a.m. And I'm looking at this list, and there's some people who are failing attendance. And because it's midterm, I'll remind you that you can go back and watch the videos and at least get half credit. As a matter of fact, I recommend that, not just for the points, but if you've not seen a single lecture or like 3 or 4 of them, then what I'm about to tell you when we do this lecture probably won't make sense because a lot of this is cumulative knowledge. So, like I said, halftime's over, so to speak. We're going into the third quarter. Be ready to get some work done. Let me help you. I would say, generally speaking, and this is not always, but generally speaking, historically, people who whatever grade there is at midterms generally doesn't change much. There are some people, a lot of people who have midterms at the F or at F at the midterms and then an F at the end. And then like the last weekend, they attempt to get some work done. But generally speaking, that's how it works. So please let me help you if you need help. I'll be sending out emails later, like official through Achieve or whatever it is through WVSU. We've got to log not only your attendance, but your grades. So you'll be getting those official emails, and I'll list the ways you can improve your grade if you need it. A lot of you don't need it, so no worries, right? Anyway, point is, again, third quarter, right? We're done with it. We're halfway past the semester, so please let me help you get caught up. Now's the time to worry about it. We have online courses that are 8 weeks long, for example. So that semester just ended for the online students. And I don't worry about it. But so for the online students, right, like it happens, it always happens. So this was their last weekend. And I had 2 students who just turned all the assignments in at the end thinking that they were going to, pass, but they couldn't. Like I can't grade everything if you turn it all in at once. So again, we're halfway done with the semester. let's get to work. Let me help you if you need help. That's what I'm here for. Get a tutor if you need it, but I think I'd be better than the tutor because I know what I'm going to test you on. The tutor doesn't. So please let me help you. Anyway, any questions before we do attendance? All right. So if you're online, again, you have to send these words in. And as I'm calling out names, if you don't hear your name online, don't worry about it. You don't need to let me know because you're going to send me the words, right? So when I go through this list, if I say you're not here and you're online, don't worry about it. Just send me the words at the end and you'll be okay. So Abigail is here. Kincaid? Not here. Andrea? Not here. Mullins is here in person. Lewis is here in person. Cooper is here in person. Hunter is here in person. Caleb's here in person. Crowder is here in person. Reinhardt is not here. Isabel is here in person. Julie is online. Well, was online. Julia? Yeah, Julie's online. Talbert, not here. Kennedy is here in person. Hannah is online. Larissa is here in person. Madeline, I think, is here. Yep, in person. Hughes is online. Galloway's online. Cook is here in person. Oh, Galloway's here too. You're both. You're everywhere. Omnipresent. Galloway, let me make sure I've got this. Galloway is here in person. Got it. Okay. Cook is here in person. Morgan's online. Tate is here. Burton's online. Kinder? No Kinder? Okay. Jones is here. Canterbury, not here. Hobson, not here. Fisher's online. Is there anybody? Andrea? Anybody else I missed? All right. What else for reminders? Since it's been a week. What else other than midterms? But again, I want to reiterate, some of you are really in danger of failing and need a lot of work, but you're not alone, right? So let me help you do it. I'm going to send you emails. Wait for the email. If you're really in danger of failing, I'm going to send you a long email that's detailed. Like, first thing you need to do is this, and then you need to do this. So please let me help you. It's not too late. What else for reminders? You should have 70 points of independent work by now. So that means what? We've got 3 weeks left until you should be done with it. So make sure you get that done. Lab, I think this week's lab is the last one as a team effort. So moving forward, if I remember correctly, the rest of them are online. So yeah, we'll still meet as we usually do, but they won't be team efforts anymore. Everything will be virtual, right? Because how are we going to have a lab about evolution? We can't just make things evolve and watch them evolve, right? It takes generations for evolution, for example. Speaking of lab, make sure you've done your pre-lab and the video. So if you haven't done that yet, might as well just come on Wednesday if you can. If you haven't done the pre-lab and the video, you can't come to lab. So make sure you do that. What else? I think that's it. The exam. Oh yeah, the exam is due by Friday, right? So it's been a week. We got one more week. Any questions about that? And even though the exam has started, we can still do an exam review. So if anybody wants to meet me online and go over the study guides, which I use to write the exam, let me know. I'd be happy to do that. What else to say? Okay, I think that's it. Oh yeah, and then the exam, like I mentioned earlier, the last time we met, the questions are worded oddly because people cheat. Not necessarily you guys, but people before you have cheated, so I've got to word them very weirdly. So ignore that. When you see something where there's words in red and words in black, forget the words in black, just focus on the words in red. It'll literally say, answer the question in red, ignore everything in black. And there'll be a bunch of questions in black, a bunch of weird nonsense. And then there's be a question in red, and that's what you need to answer. So when you get those questions, to save you time, just focus on the red questions, ignore everything else. You guys good with that? All right. And just as a reminder, it is a timed exam. It's 50 minutes because it's supposed to be closed book. So if you want more time, just email me. We'll find a time to meet online. If we can meet online and I can see you and I can see your screen and I know you're not, using notes or books or whatever, I don't care how long it takes you. Take you 3 hours. I don't care. But as long as I can ensure you're taking a closed book exam, I don't care how long it takes you. The only reason it's timed is to ensure that no one's using notes and things like that. So any questions about the exams or independent work, labs? I think that's it. All right. Oh yeah, the last thing. I'm really trying to give the TA some experience with grading. So you'll notice some of the labs are slow to get back to you with grading. And I've been doing a bunch of it. I'm really holding off on grading the photosynthesis respiration lab for some of you, or in general. The good news is, though, the longer it takes for you to get it back, the longer you have to do the lab report, right? Because it's not due until 2 weeks after you get your lab back. So on one hand, I apologize that it's taken so long to get those labs graded. On the other hand, if you haven't had it graded, focus on the positive, I guess. Any questions about anything before we get back into this? All right, so to remind you, we're finishing up this discussion about meiosis. We almost finished this chapter on the Friday before spring break. It would have been perfect, but we didn't quite get there. So really quickly, let's finish this chapter and jump into the next one. So the last thing we were talking about was non-disjunction during meiosis, which is a situation where, in meiosis, the homologous chromosomes separate, then the sister chromatids separate, and sometimes there's problems, with that, right? Sometimes things go wrong. The last thing we were talking about was what happens when you have non-disjunction with sex chromosomes, and that's what this was, right? So you have this example up top. Actually, that'll be the first official word for attendance will be sex, since we're talking about it. But yeah, so you can see here, that's an example of you have an extra X chromosome. This one has an extra Y chromosome. This one has 3 X chromosomes instead of 2, so on and so forth. You don't need to know this. This is just examples. You can read it if you want. If anything, there might be some extra credit questions, but I don't require you to know anything about these syndromes. It is a decent topic for independent work, though, if you want to look into it. Obviously, it gives you the origins, it gives you the symptoms, but how common it is, but you can look into it deeper if you want. So let's see. Here's another thing that won't be on the exam, but it's definitely worth mentioning. Right, so not only do we have problems sometimes with the chromosomes separating as they should, but sometimes we have damaged chromosomes, right? Sometimes there's partial duplications and deletions, right? So when we go through this process of chromosome duplication in S phase of interphase, sometimes it doesn't work the right way, right? Sometimes they don't completely or correctly duplicate. Sometimes things get deleted. And that's usually survivable, but there's usually physical and mental abnormalities. Then there's something called inversions, which is when the chromosomes or sister chromatids become attached. They have a 180-degree rotation, meaning they turn all the way around, and then they get reinserted. And that's usually a mild effect, and I'll tell you a little bit more about that on the upcoming slide. Then there's something called translocations, and that's when the part of the chromosome disassociates, right? So it completely comes off, and then attaches to a whole different chromosome. Right, so as you might imagine, that causes some big problems too. And also, imagine this, and again, I'm not going to test you on this, but it's worth thinking about. If any of this stuff happens to somebody, and they can still survive, and they can still reproduce, then that means all their descendants are going to have that too, right? It's there forever. Right? Anyway, again, that's just extra stuff. I'm not going to test you on that. If anything, and probably not even this, you might get some extra credit questions on that. So, any questions about those concepts of damaged chromosomes? All right. And again, good independent work topic if you want. So last time we spoke, the independent work topic I suggested was, the different disorders if you have extra chromosomes, right? We talked about trisomy 21, which is having an extra 21 chromosome. But what about the other ones? What happens if you have extra other chromosomes? What happens if you have too few of the other chromosomes, right? And now I'm suggesting this. What are some disorders, right? I haven't told you any of the disorders. I just said these are some of the things that happen. Sometimes it's really bad. Sometimes it's mild. But there are names for it. And if you want, you can look it up for independent work. Here's a picture of an inversion, right? So this is a homologous chromosome. And this is the way it should be, right? But instead, it kind of flips itself, right? This is a reciprocal translocation. So here we have this chunk of chromosome being swapped with that chunk, right? In a sense, kind of, I'm really stretching it here, but in a sense, that's a little bit like crossing over. Remember, in crossing over, you have two homologous chromosomes, one from your mom, one from your dad. They're in close proximity, so they swap chunks. This is similar to that, except now we're not talking about homologous chromosomes here, right? These are homologous chromosomes. These are not. So that would be more of a problem, generally speaking. Anyway, any questions about that? All right. So this is from your old textbook. I just included it on the PowerPoint just in case you want to read it. I'm not going to test you on this either. But what I liked about your old textbook was, or the old textbook, is that at the end of every chapter, they had what's called the evolution connection. And every topic, every chapter they covered, they somehow brought it back to evolution. So anyway, you can read that if you want. The advantages and disadvantages of sexual versus asexual reproduction. We will talk about that when we talk about evolution. But for now, that's an introduction to it. You can read it if you want. And that's it for that chapter. We'll jump into the next one. Are there any questions about mitosis, meiosis, cell cycle, cancer, any of that? All right. Let's jump into the next one. Actually, and that'll be the next word for attendance. Next. We're jumping into the next chapter. So again, to remind you, this is only the second semester I've used the new textbook. I like the old textbook in some ways more than the new one, but I also like the new one in some ways more than the old one. So I like to merge the two, and you'll see the same thing here. So if you're following along closely in the textbook, you'll notice that in this chapter I'm kind of skipping around, right? So I'm following the order that the old textbook used. So it's the same information, just kind of presented in a different order. So let's talk about patterns of inheritance. Basically, this chapter is about genetics, which to me is more fascinating than anything we've covered so far. To me, this is where it starts getting interesting. Why genetics matters, again, this is from your old textbook, but let's talk about this. So what's this on her arms? You guys know what those are? What are those? Tattoos, right? She has tattoos. Now, if she were to have a kid, do you think that kid would be born with tattoos? Smart man. He is correct. That kid will not be born with tattoos. Do you think that kid will be born with that color hair? I would guess not, because in my opinion, it looks like she colored it, right? The point being here is humans and every other living organism, they change as they're alive, right? Things happen to them. Sometimes they change themselves, like she did. I've got buddies who have lost a leg because they got blown off in a war, but when they have kids, they came out with two legs, right? Point being here is when we talk about genetics, again, there are things that happens to an organism, but unless it's heritable, it doesn't matter, right? So getting a tattoo doesn't matter for heredity because that's not heritable. Right? Getting your hair colored doesn't matter for heredity because it's not heritable. Getting your leg blown off, it's not heritable. All these things, right? So that's a concept we'll come to later in the chapter. Here we go. This is a father holding his little baby daughter. Any idea? Well, there's a million things, but any idea what the textbook may have been getting at with that picture? Because they have a point. And I don't blame you if you don't get it, because my mind would go in a million different directions with this. But any guesses what they might be trying to get at there? It's okay. So what the textbook is getting at, right? So that's again a father with his daughter. It's because the father determines the sex of a child. I mean, not on purpose. It's not like the father says, "I'm going to have a girl," and pushes out the right sperm. But the mother, right? If you're a female, your eggs. You only have X chromosomes, right? All your cells, you only have X chromosomes. So the only thing you're offering is an X chromosome. The male is either offering an X chromosome or a Y chromosome. Therefore, it is the male. That sperm determines the sex. So that's something we'll talk about later. And then it's just a picture of a family, which is great because you can see, again, when looking at ancestors and descendants, you can see similarities but differences. And these are all things that we talked about with the last chapter, talking about sexual reproduction. How you combine chromosomes, combine genes, combine traits, and that's all they're getting at there. So anyway, let's jump into it. Obviously, also, sorry, to answer the question, that itself is a good independent work question, why genetics matters. You can ask yourself that and look it up, but for a lot of reasons. Cancer, some people are at higher risk of cancer and they can pass that along. Heart disease, dementia, a lot of things are heritable, right? So genetics is very important. In some cases, it's a matter of life and death. Again, from your old textbook, they always started off the chapter with a sort of like an interesting story, if you will, something that's more interesting than the rest of the chapter. Because unless you're a biologist, you probably don't care about everything I'm about to teach you, even though it's important. So your old textbook tried to give you something interesting. You can read about it if you want. Talks about dogs and artificial selection. To me, it's interesting. I'm not going to waste my time talking about it, but I kept it on the PowerPoint just in case you wanted to read it. And again, great independent work topics if you want to look into the genetics of dogs. There we go, genetics and heredity. So once again, I'll remind you, I'm teaching all the information that is in your textbook. I'm going to teach it, but I'm doing it in a different order, in a different style. I'm doing this chapter like it was the other textbook. And what I really liked about the other textbook in this chapter was that it kind of told this as a story, which to me is easier to follow, right? To me, sometimes stories are easier to follow than just like, here's a fact, here's a fact, here's a fact, right? So let's talk about it. All right. Well, we'll get there. The first thing you need to know is the word heredity. That is the word I might test you on. That is definitely a word that I'm going to use, so you need to know what it means. That is the transmission of traits from one generation to the next. Transmission of traits from one generation to the next. That is the biological definition of heredity. Right? It'd probably be different in legal terms, right? Heredity in legal terms probably has something to do with property, something like that. So any questions about heredity? The definition, that is. All right. Genetics, then, is the scientific study of heredity. I'm almost definitely not going to ask you what the definition of genetics, because this whole chapter is on genetics. There's going to be a lot of questions about genetics, but not the term genetics. Gregor Mendel is somebody we consider the father of heredity. I'm not going to ask you any questions about him, but like I said, your old textbook kind of told this like a story, and he's kind of like the main person of the story, so I'm going to use that. But most likely, there's not going to be any questions about him. I'm certainly not going to ask about the time frame, but yeah, it was in the 1860s when he did his work. But he argued that parents passed down their unique genes to offspring. So this was new back then. I mean, even back then, they could tell, this parent and this parent get together. Kids look like a mixture of those parents. Like, we knew that. But what we didn't know was there was things called genes. Of course, he didn't call them genes, but I use that word because that's eventually what we came to call them. So he knew there was these things, what we now call genes, and he knew that they were getting passed down to their offspring, right? That was actually groundbreaking information at the time. He knew that these genes, and again, he didn't call them genes, but whatever, he knew that they were responsible for these inherited traits. So, looking at, I don't know, two parents with brown eyes have a kid with blue eyes, for example, right? He knew something was up with that. We'll come back to that. Actually, do either of you have blue eyes who have parents who have brown eyes? Yeah, just one of you? Me too. Both of my parents have brown eyes, and I have blue eyes. We'll talk about that later. He also argued that genes retain their individual identities generation after generation, no matter how they're mixed up or temporarily masked. Again, using the blue eyes as an example. Both my parents had brown eyes. Both her parents had brown eyes, but we have blue eyes. So those genes were still there. They were just temporarily masked in that generation. And then they showed back up in my generation and in her generation. So I would say if you're taking notes, I guess there's not necessarily anything to write down here. This is kind of like a prelude. This is what he argued, and these are all the things that we're going to talk about in this chapter, amongst other things. But are there any questions so far? All right. The next word for attendance will be garden. As a reminder, because it's been a while, spring break, so we're coming back. So as a reminder, if you're here in person, you don't have to send those words, but you can because they're extra credit. If you're watching this online, live streaming right now, you have to send those words before 9 a.m. That's how you get your credit. And if you're watching the video, again, you'll need to send the words in addition to whatever other instructions are on that document. Anyway, so here's what we're going to talk about. First, we're going to talk about what happened in Abbey Garden. Because again, like I said, your old textbook tells us like a story. And so I like this better. We're giving you the location. It's like the movie is taking us to Abbey Garden. That's where he did his work. what did he work on? He studied peas. I'm not going to ask you that either, but really quickly, I'll tell you why. He studied them because they're easy to grow, right? So, for example, if you're studying genetics, you probably won't want to study a redwood, right? Those things are huge, hundreds of feet tall. You know, if you were to plant one, you'd be dead before it grew up, right? So that wouldn't be good. It's not easy to grow. And also because they come in many readily distinguishable varieties. So again, you know, you wouldn't want to use grass. Well, that's not a good example. Maybe dandelions, right? You wouldn't want to use dandelions. Dandelions are easy to grow, but they don't come in many readily distinguishable varieties, right? They just look the same. You know, those spiky leaves, yellow flower that turns into a white puffy thing, right? There's no variety there. But anyway, he studied peas. I'm not going to ask you about it, but we're going to be talking about peas a lot because that's what he studied. Two terms you definitely need to understand are these right here. Make sure you understand these. I might test you on them, but I'm definitely going to use them. The whole lab this week is based on these terms, sort of, kind of. Character is a heritable feature that varies among individuals. Hair color is a character. Eye color is a character. Height is a character. Widow's peak is a character. Having freckles or not having freckles is a character, right? And that brings us to trait. A trait is a variant of a character. So again, like I said, eye color is a character. So blue eyes would be a trait. Brown eyes would be a trait. Green eyes would be a trait, right? Those are all traits of the character eye color. Does that make sense? Make sure you understand those concepts, those terms, and how they're related to each other because we will be using them a lot. Each character that Mendel studied when he's working on these pea plants occurred in two distinct traits, which is important. It's also worth noting, I'm going to mention this later, that most things are not like that, right? So yes, that's what he studied. He studied things in two distinct traits, but a lot of the things are not in two distinct traits. So the lab we're doing this week, somebody chose things that came in two distinct traits, right? You either have a widow's peak or you don't. You either have a cleft chin or you don't. You either have, I don't know, hitchhiker's thumb or you don't, right? All those things. Hopefully you know what I'm talking about because you've done the pre-lab already. But yeah, the things he studied did come in two distinct traits, which is important. That's otherwise he wouldn't have been able to do his work. But also keep that in mind as I'm going through this, as we're learning this, keep in mind that most things don't come in two distinct traits. So it's almost like I'm teaching you this, I don't know, easy version of reality. And then once you've learned the easy version, then we'll come back to it and talk about the actual reality. Anyway, let's move forward here. So another important advantage of pea plants is that he could strictly control the reproduction. So I'm going to go through this very quickly because, again, I'm not going to test you on this. But long story short, he was in control of the reproduction, right? So, for example, you wouldn't want to study, I wouldn't say that necessarily, but one of the disadvantages of studying humans, if you're going to study genetics, is you can't control the reproduction, right? You can't say, hey, tall person, I want you to have sex with that short person so I can study your children, right? That's unethical. But with pea plants, you can do that. Anyway, some things really quickly, just so you know. Again, it's not important because I'm not going to test you on it, but some plants require fertilization from other plants. Pea plants can fertilize themselves if they want. So you've got the male parts and the female parts all enclosed into the same flower. So really, that could impregnate itself, right? So to speak, could fertilize itself. The sperm from that flower could fertilize the egg from the same flower. So what he did was basically he cut off the male parts, right? So he could cut off the male parts to ensure that the pollen, which contains the sperm, didn't fertilize that particular flower. So once again, long story short, Mendel was in control of what mated with what, right? So he could say, all right, I want this plant to mate with this plant, and then I'm going to study the offspring. But again, I'm not going to test you on this. This is all kind of an introduction to what we're about to learn. One term you do need to know is this one, though, purebred. Purebred varieties of plants self-fertilize to produce offspring that are identical to the parents. So you need to know that. And this will make a little bit more sense as we go further into this discussion. So, for example, we'll talk about, flower color is one of those characters that we're going to look at. Because there's two traits. It's either white or purple, right? So a purebred white pea plant will produce offspring that have white flowers. A purebred purple pea plant will produce offspring that have purple flowers, right? So he had these purebreds that he knew whatever trait he was looking at or whatever character he was looking at, he knew that if he were to self-fertilize that plant, that the offspring would have the same characters, the same traits. But when he crossed two different purebreds, that was something called a hybrid. You need to know that too. Not that he did that, but you need to know what a hybrid is. And a hybrid is an offspring of two different purebred varieties. So make sure you know what a purebred is. Make sure you know what a hybrid is. And there's a bunch of other words we're going to talk about really quickly, but these are a little bit easier in my opinion. So like I said, a hybrid is a cross between two purebreds, but the actual act of crossing them is called a genetic cross, right? That cross fertilization. So a genetic cross produces hybrids. The parental plants are called the P generation. So again, in this context, if we said we had this purebred purple pea plant and this purebred white pea plant, right? That would be the parental plants. Then you do a genetic cross to form your hybrids, right? And those are in the F1 generation. So you have the P generation. They have children. Those are called the F1 generations. And then if you were to cross that F1 generation, then those offspring, the offspring of that would be the F2 generation. So in a sense, you might think this is like, I don't know, great-grandparents, grandparents, parents, and kids. In a sense, as loosely defining it, but that is kind of how it's related. Does that make sense, everybody? So we have the purebred plants. We cross them to make the first generation, which is the F1 generation, and then we can cross them to make the F2 generation. I should also point this out because neither your current textbook or the old textbook talked about this. But this is how he did things, and this is how a lot of people still do things. But it's also a lot more complicated than this in real life. So sometimes you might, for example, take a plant from the P generation and cross it with a plant from the F2 generation. Or you might cross something from the F2 generation with the F1 generation. Then, of course, there's F3 generations, so on and so forth. So this is really just a Cliff Notes version. So keep in mind, everything I'm about to teach you, while it's true, it doesn't represent everything that happens in genetics. It is actually a lot more complicated than what we teach you. Any questions so far? All right, let's talk about the law of segregation. And you've already been slightly introduced to this. When we talked about mitosis and meiosis, you were slightly introduced to this concept. So let's jump into it some more. So Mendel performed experiments that tracked the inheritance of characters, for example, flower color, and some other ones that I'm going to tell you about later, that occurs as two alternative traits, right? So we've already said that. There's nothing new so far that I've said, right? That's a reminder that he studied characters that had two distinct traits. So for example, if he was studying humans, he wouldn't study height, right? Because it's not like all humans are either 6'6" or 5'6", right? We're everywhere in between, and also taller and shorter, right? There's not two distinct traits for height for plants too. So what he looked at were things that had two distinct traits. In the next slide, we'll show you some of them. And the results led him to formulate several hypotheses about inheritance. We'll talk about that after this slide. But here's what I was talking about, the two distinct traits. If you're studying, don't study this. You don't need to know this. But yeah, these are some of the things we'll talk about, especially that for some reason, almost every textbook you read, they really focus on this, the flower color. I think probably because that's just the easiest to see and visualize. But yeah, so the flower color is either in purple or white. The flower position's either at the end or not at the end, right? The seed color is either yellow or green. Or that W is trying to escape. But anyway, the seed shape is either round or wrinkled, so on and so forth. So those are the characters he looked at, because again, there's two distinct traits, right? Any questions so far? All right. Actually, let me use this time. Let me make sure this is working the way it should. Haven't heard anything. Okay, good. I remember Friday before spring break, something was not working and it wasn't presenting, so I wanted to make sure we were good there. Anyway, let's jump into it. The monohybrid cross. This is a term you need to know. Probably not necessarily going to ask you what a monohybrid cross is, but you need to understand it because I'm going to use the term. A monohybrid cross is a cross between the purebred parents, which is basically all the crosses I'm going to talk about. I don't want to say that either. Never mind. But anyway, A monohybrid cross is a cross between two hybrid parents that differ in only one character. So backing up, if we were to cross plants that had a white flower and a purple flower, then he would make sure all the other things are the same, right? They would have the same flower position, the same seed color, the same seed shape, the same pod color, the same stem length. The only way they would be different was the flower or the color, right? Because he just wanted to study that one particular character. And when he did that, for example, again, crossing a purple flower with a white flower, purebred purple flower with a purebred white flower, all of the offspring had purple flowers. And think about it. Now that's not really mind-blowing, but at the time that was probably pretty interesting. Like, why? Why is it that every time I cross these purple flowers with these white flowers, I always get purple flowers? What happened to the white flower gene? Of course, they didn't call them genes back then, but that was the question, right? So that was the observation was, again, across these two purebred varieties, and we always have purple flowers. That's the question, or the observation. The question then is, what happened to the white flower, right? What happened to that gene? And he found the answer because he took that F1 generation, right? So again, picture it. The P generation was all purple flowers and white flowers. He crossed them thousands of times, and every single time he did it, without exception, 100% of the offspring had purple flowers. Never once was there one with a white flower. So then he took those from that generation and crossed them, and then he found out this. He found out that about 75% had purple flowers and about 25% had white flowers. And I like using those words because I don't think this textbook or the old one really focuses on this fact. This makes it look like Mendel took one purple flower, crossed it with one white flower, had one plant that had purple flowers, crossed it, and then had 4 plants, 3 of which were purple, 1 was white. That's not the case. This is an oversimplification. He did this hundreds, maybe thousands of times, right? But the part that's not an oversimplification is this. No matter how many times he crossed the purebred purple with the purebred white, no matter how many times, 100% of the offspring were purple, right? And then this is where it got a little bit iffy, right? So again, thousands of times he crossed those generations and then got about 75% purple and 25% white. Make sense? I'm going to pause for a second. That is a ratio you need to know for a monohybrid cross. If I have any questions, let me think about how I'm going to word this. Whenever you cross an F1 generation, well, you know what? We'll come back to it. But yeah, we're going to come back to this ratio. That is a ratio you do need to know, but I'll explain it more later. Because what we're going to talk about later is how we went from this to this, right? We're going to look at the chromosomes that cause this. Like what's going on at the cellular level that's causing about 75% of the flowers to be purple and 25% to be white. So also, again, before we move forward, I just want to remind you that this is just an example. You don't necessarily need to know anything about purple flowers and white flowers. And also remind you that he did other things too. It wasn't just purple or flower color, right? There was seed color, seed shape, flower position, all those other things. But these were always his outcomes. Again, 100% purple and about 75% purple, 25% white. So what he figured out was the gene for the white flower did not disappear, right? Which, again, at the time must have been very interesting, right? So, again, he did it hundreds, thousands of times when he crossed those purebreds, always got purple flowers, right? Which kind of made sense a little bit. But then probably when this happened, that must have been a little bit weird. Like, why do it when I crossed then these purple flowers, other than, not the hybrid one or the purebred ones, why is it that when I cross these that we get about 75% purple and about 25% white? So what he figured out was that those F1 plants had 2 genes. Well, I'm using the word genes. There's a different word for it. We'll talk about it later, but for now, genes will work. There was a gene for purple flower and a gene for white flower. So again, backing up, he figured out these right here, the F1 generation, he figured, even though they're all purple, must have had a gene for purple and one for white, right? That's what he figured out. Otherwise, these offspring right here, you wouldn't have any white ones. But surely that gene for whiteness, for a white flower, must still be there hidden. Otherwise, they wouldn't have any offspring with white flowers, right? That's what he figured out. And that's what this slide basically says. So any questions about that? All right. From these results, he came up with 4 hypotheses. And you need to know these 4 hypotheses. First, there are alternative versions of genes that account for variations in inherited characteristics. Alternative versions. This is what our whole lab this week is based on, is these alternative versions of genes. So you need to know that. Of course, at the time, for him, these were hypotheses. We know this now. It's not really a hypothesis. It's more like a theory or even a law. These are just things we know now. There are alternative versions of genes. These genes, or these alternative versions, are called alleles. You definitely need to know that. Alleles. So again, using eye color as an example, both my parents had brown eyes, but I have blue eyes. So I know that obviously they had alleles for brown eyes, otherwise they wouldn't have brown eyes. But they also carried an allele for blue eyes. Otherwise, I wouldn't have it, right? I got it from them. Also, I've done a 23andMe, so I know I'm not adopted. Number 2, for each inherited character, an organism inherits 2 alleles, one from each parent. You definitely need to know that too. And so far, that's not very different from what you learned in the previous chapter. In the last chapter, I told you that there's things called homologous chromosomes. And you got one version from your mom and one version from your dad, right? You have 23 pairs of chromosomes in each of your cells, well, other than your sperm and your egg. And for each of those 23 pairs of chromosomes, you've got one from your mom, one from your dad. This is the same concept, except now we're being really specific, and we're talking about a specific gene. So the gene for chin dimple, right? You got one from you have an allele from your mom, you have an allele from your dad. Any questions about these 2 so far? Parent will be the next word for attendance. So again, if you're in person, you don't need to send those words, but you can for extra credit. If you're watching this live, you need to send those words before 9 a.m. to get your credit. So, some other words you definitely need to understand. If an organism has 2 of the same alleles, they are homozygous. Homo meaning same, right? So like when you think, I think probably the most common word I think people associate with homo would be like homosexual. You're attracted to the same sex. This homozygous means you have the same alleles. For me, for example, or anybody in here, if you have blue eyes, you are homozygous. You have 2 alleles for blue eyes. And I'll explain that later. So yes, 2 identical alleles is homozygous. 2 different alleles is called heterozygous. Again, using sex as an example because most people are familiar with that term. A heterosexual is somebody who is attracted to a different sex. Heterozygous means they have different alleles, right? Homo, same. Hetero, different. Any questions about those 2 terms? Those are very important terms. There will be questions. Yeah, there's going to be at least one question when I ask you to not identify, but man, what's the word I'm looking for? I can't think right now. Anyway, you will need to know those 2 words, and there's going to be other questions where I'm going to use those words in the question. Like, so you have to figure something out, and to be able to figure that thing out, you need to understand those words because I'm going to be using them in the question. Define, that's the word I was looking for. Yes. So yes, there will be at least one question where you have to define homozygous, at least one question where you have to define heterozygous, and then other questions where I say a person homozygous for this had sex with somebody who is heterozygous for this. What are the chances that their kids are going to have that? Right. So there's going to be questions like that, and you're going to need to understand what homozygous and heterozygous mean. Otherwise, that question won't make sense to you. So any questions so far? All right. Oh yeah, heterozygous. I don't find these memes, or I don't make them. I just find them. All right, this one too. I didn't make that meme either. Anyway, the third hypothesis of his is if the two alleles are different, so in other words, if somebody's heterozygous, then one of them determines the appearance and the other one has no effect at all. The one that determines the appearance is dominant and the other one is called recessive. This is how I'm going to teach it at first. Just so you know, later on, I'm going to tell you there's exceptions to the rules. So just know that now. Everything I'm about to teach you is there are exceptions. As a matter of fact, this really is the exception. But because it's the simplest way to teach it, everybody teaches it this way first. So we're going to teach you this simple version. And then once you've learned that, then we'll talk about the way things are mostly in real life. But yes, you definitely need to know this. Dominant, recessive, right? Dominant, recessive. So if we're talking about two different alleles, at least for now, one of them is always dominant, one of them is always recessive. So if one of them is dominant, the recessive one doesn't even matter. So again, like my parents and her parents, they had an allele for a brown eye and an allele for a blue eye. They had brown eyes because the brown allele is dominant. The blue allele is recessive, right? As long as that dominant allele is around, the recessive allele doesn't matter. Another thing, and you'll be really familiar with this after lab, is that when we write them, uppercase is always dominant, lowercase is always recessive. So, you know, this probably won't work. This doesn't draw very well, but, you know, yep, didn't draw. A homozygous dominant Q, whatever gene that may be, would look like big Q, right? Heterozygous would be big Q, little q. Homozygous recessive would be little q, little q. Anyway, that's just an example. Any questions about dominant versus recessive and how we write them? All right. Finally, the fourth and final of his hypotheses. A sperm or egg carries only one allele for each inherited character. And you already know that, right? Except in the previous chapter, we didn't say allele or gene. We were talking about the whole chromosome, which is where these alleles are, right? These chromosomes are on the chromosome. These genes, these alleles are on chromosomes. So now we're just being specific and we're talking about these chromosomes, these genes. But yeah, what we said here, you already knew that from the last chapter. But now we're just saying in a different context. Because the reason they're inherited separately is because the two alleles for a character segregate during meiosis, right? When you produce gametes, when you produce your sperm, when you produce your eggs during meiosis, they segregate, right? So again, using my parents as an example, just to keep it consistent, they both had brown eyes, right? So my dad had brown eyes. He had an allele for blue. He had an allele for brown. When he produced sperm, they separated, right? So 50% of his sperm carry the allele for blue eyes. 50% of his sperm carry the allele for brown eyes. And this is called the law of segregation. So make sure you know that. There will be a test question on that. There's a question on the study guide about it. You need to know what the law of segregation is, right? So the law of segregation is basically saying that, yeah, you have two alleles and they segregate when you produce sperm or egg. If you're talking about the alleles, you could also say the same thing if you're talking, having this discussion, but in the context of chromosomes. So in the context of chromosomes, the law of segregation states that one homologous chromosome goes to one of the gametes and the other one goes to the other. They segregate. And then, of course, as you learned from last chapter, and probably as you knew your whole life, when the sperm and the egg combine, right, when the sperm fertilizes the egg, then each one contributes its allele, and that restores the pair condition. So again, my dad has a blue allele and a brown allele. When he produces sperm, 50% are blue, 50% are brown. My mom's the same way, right? When she produced egg, 50% were blue, 50% were brown. And then during fertilization, by chance, my mom produced or gave an egg that had the blue allele. By chance, my dad gave a sperm that had the blue allele. Now here I am. Anyway, it's a very important concept. The next slide I'm going to show you visualizes this, and then I'll see if you have any questions. But this is the law of segregation. So it's the same thing I've already shown you, except now they're showing the alleles, right? So what we're looking at here, and I think this will be better. The old textbook made a mistake, in my opinion. What we have here, oh, no, they did do it. I'm sorry. So, right, that's homozygous dominant, homozygous recessive. right? And so when this thing up here we go. When this thing goes through meiosis and produces sperm or eggs, they separate, right? But because they only have big P's, then even though they do separate, still 100% of the sperm or egg is big P, right? Same thing here. These are going to get separated, segregated during meiosis. But because the only thing it has to offer is little p's, then still 100% of the sperm or egg are going to be little p. Now, because of that, when you cross these things, there's a 100% chance you're going to have a big P mixed with a little p, right? Which gives you the heterozygous condition. Big P, little p, right? Dominant and recessive. That's heterozygous, the two different alleles. One dominant, one recessive. 100% of the offspring had that. Then this thing goes through meiosis and it produces sperm or eggs. And when it does that, again, they separate. So 50% of the sperm or egg are going to be big P. 50% are going to be little p. And then, of course, when you cross those, this is where things get a little bit more complicated, but we'll come back to this. I'm not going to take questions yet. We have this. We can then figure out the chances of the offspring, right? So when you were to cross these, you would figure about, like we said earlier, here's a ratio that you should know. About 25% are going to be white and about 75% are going to be purple, right? Or 3 to 1, if you want to look at it that way. Three purple to 1 white. That's what we expect when we look at them. That's the phenotype, and that's a term I'm going to come back to here shortly. But the genotype, which is another term I'm going to come back to here shortly, is also different, right? So here we have a homozygous dominant. Sorry, I forget the people on the screen can't see me. Here we have a homozygous dominant, right? Big P, big P. Here we have two heterozygous, right? Big P, little p. And notice new, by the way, they have different genotypes, right? Again, this one's homozygous dominant, the top left, and the other two are heterozygous, but nonetheless, they're still purple. And of course, at the bottom right, then we have one that is homozygous recessive, little p, little p. So these are all things you're going to need to know later. Right now, I'm just introducing you to it. But later, you're going to need to be able to do this yourself. In the exam and in the lab, I'm going to say this thing crosses with this thing. What do the offspring look like, right? So you're going to need to be able to do this. And a little bit later, you're going to need to know this ratio, right? A 3 to 1 phenotype ratio, a 1 to 2 to 1 genotype ratio. These are all things I'm going to teach you later. Same with the terms phenotype and genotype. These are all things I'm going to teach you later. Are there any questions so far? We're almost done for the day. So there's these things that I just showed you. They're called Punnett squares. You're going to need to do Punnett squares in lab today or this week. The Punnett squares gives us the 4 possible combinations of gametes, which then obviously gives us the 4 possible combinations, or excuse me, the possible offsprings that you could have. And each square represents an equally probable product of fertilization. So again, like, you know what I mean? Like this, if you were to cross this plant, right? These heterozygous plants, there's a 25% chance that the offspring are going to be homozygous dominant. There's a 50% chance that they're going to be heterozygous, because there's 2 of them, right? 2 out of 4. There's a 25% chance that they're going to be homozygous recessive. That's a concept that we're going to come back to. It will be really important later. Yeah, so when we get back, we'll talk about this. And the last word for attendance is makeup. You guys keep an eye on your emails. Like I said, it's midterms, so I've got to put grades in. And then not only am I going to put in grades, I'm going to send you the official email to tell you how we can help your grade and get you back on track if you need it. Most of you are on track and you're doing okay, but some of you need it, and I'm here to help you. And that's it. I'll see you either today in lab or Wednesday. And again, if you didn't do the pre-lab, don't come to lab. Just show up on Wednesday. It's okay if you show up on Wednesday, but you cannot come to lab if you didn't do the pre-lab, including the video. There's a video, then the pre-lab.